Okay, today I posted homework on Connect for chapter 6 and 7. I'll work on 8 and 9 tomorrow, and at least 8 will be done. The second exam is scheduled for the middle of next week, and it will cover chapters 6 through 9. Now, today, if I remember correctly, I had through chapter 8, which is a fairly short, fairly simple chapter. It's about chromosome number, different kinds of organisms, and chromosome architecture, and kind of chromosome abnormalities that might be tolerable, and position on the chromosome genes and things like that. So basically, this is an image of the human chromosomes. And you can see they're of different sizes. They have different staining patterns. And if you know what you're looking at, you can look at these images and tell what chromosome you're looking at. Now, at least some point in the cell cycle, you can view the chromosomes in a cell with a microscope. They're not great, but you can actually see them. And the term genetic variation can mean a lot of things. But in this chapter, it's going to mean basically differences in allele frequency and chromosome number in either members of the same species or between members of different species. And you can use the term allelic variation, which is changes in the allele frequencies of specific genes. Or you can say there's variations in chromosome structure and number. And these changes in chromosome structure and number, almost by definition, are going to affect more than one gene. They can be very important forces in evolution. They can cause all kinds of diseases. And they might be important in new strains of crops. And this is especially true for plants. Now, chromosomes can be seen by light microscopy. It's not the world's easiest thing. And it is going to take practice, years of practice, to get good at this. And you can call someone a cytogeneticist because primarily a cytogeneticist is going to be a scientist who looks at chromosomes with a light microscope. Now, electron microscopes might be used for a more up close picture, but very powerful light microscopes can actually pick these things up. And every species has a defined chromosome number, and each chromosome of every species has a defined size. And okay, this is just a picture of chromosomes in a human. There's from a fruit fly. There's from a corn plant. And the individual chromosome makeup will be fairly unique to each species. And so you can classify chromosomes in a variety of ways. You can order them by size, and chromosomes are usually just given numbers. And chromosome number 1 is the biggest, and then number 2 is the second biggest, and so on and so forth. If the species in question has sex chromosomes, they are giving usually letter designations. They could be called X and Y, or in certain other species, non-mammalian species, they might be called W or Z. And then there are some species, animals and plants, that don't tend to have sex chromosomes, but they do have opposite sexes. And so sex determination is determined in some other fashion. Every chromosome will have a centromere, which is more or less a little position somewhere but down the length of the chromosome. And this centromere might be more or less in the middle. It might be off center. It might be very off center, or it might be what they call a telocentric centromere. It's at one end. And there's a variety of ways you can stain chromosomes. And the typical chromosome will have dark staining regions and lighter staining regions. And those are called bands. And so you can describe a chromosome by where the centromere is. If you say it's metacentric, it's right around the middle. If it's submetacentric, it's off center. If it's acrocentric, that's even more off center. And if it's telocentric, it's at one end. Chromosomes are always going to be designated. They have two arms, the P arm and the Q arm. The P is the shorter arm, Q is longer. And in the case of this telocentric chromosome, the P arm has a length of 0, and the Q arm has some other length. Now, for every individual organism, for that matter, for every individual cell, you can make what they call a karyotype. And the karyotype is simply a micrographic picture of all of the chromosomes that you can extract from a cell at a certain stage of the cell cycle. And typically, you extract the chromosomes in metaphase because that is when the chromosomes are the most compact. And so that is when the chromosomes are the most visible with light microscopy. And then you have to sort all these chromosomes out according to length. And it's not incredibly difficult, other than you have to take into account that chromosomes can bend. And when these things bend, their length changes. And so you really need to use a ruler and pretty carefully measure these things out. Now, one of the ways you can stain a chromosome is Giemsa staining. And it reveals what they call the G band. And so these different chromosomes are going to have different sizes, and the centromere is going to be in different places. And the staining pattern that you observe can be used to help you identify specific chromosomes. In the human karyotype, all of these G bands have been mapped out. And you have regions of every chromosome. And so there is region 1, and then there's region 2, and so on and so forth. And all of these things have numbers. And that lets you, if you're in the know, that identifies how far away from the centromere any given band is. Now, you don't have to worry about remembering any of these things, but this is just what the human karyotype looks like after it's been stained. And so the banding pattern. Well, banding pattern helps you distinguish between different chromosomes. It can allow you to identify chromosomal level mutations, changes in the chromosome structure. And it can be used for analyzing evolutionary relationships between species. Because if the species are very closely related, evolutionarily speaking, the banding patterns will be virtually identical. And so there's some big chromosome level mutations. And a chromosome might have a deletion. big chunk of the chromosome is missing. And so all of the genes in that area that's missing are gone. It might have what they call a duplication. A big chunk of the chromosome is in there two or more times. And all of the genes in that area that's duplicated, you now have in excess. Chromosome might have an inversion. Chromosomes can break, but they can be repaired. And in the case of an inversion, they get repaired, but they're backwards. And you can have what's called translocation. A segment of one chromosome breaks off, but is attached to a non-homologous chromosome. In the case of a deletion, some DNA is always missing. And deletions are almost always deleterious. In the case of a duplication, some of that genetic material is there in excess. Sometimes that is deleterious, sometimes not. It depends upon what exactly is in more than two copies. And then you have an inversion. In an inversion, no DNA is missing. It's just backwards. And then you have a translocation. And in a translocation, no DNA is missing. It's just somewhere else. Now, inversions and translocations might cause a problem, and they might not. In your somatic cells, in your body cells, inversions and translocations might not cause any problems at all. In germ cells that make gametes, inversions and translocations can interfere with meiosis. Now, a translocation can be what we call a simple translocation. One piece of one chromosome is broken and attached to another, or it can be what we call a reciprocal translocation. Two different chromosomes have exchanged pieces of DNA with each other. And so here's a deletion. This region in between the red arrows is missing. Here's a duplication. That region in between the red arrows is now there twice. Here's an inversion. That region between the red arrows is backwards. And here's a simple translocation. This region, again, between the two red arrows, is now stuck over here. And here is reciprocal. These two chromosomes have exchanged pieces of DNA. I'll tell you when the exams are going to be due when I post them. I kind of like this idea of being due at noon. Otherwise, they're due at midnight. And if you wait until the last minute and it's closer to midnight, you can't get a hold of me. But some people say this noon thing isn't working out for them. So we'll see. Okay. Deletions and duplications. How do they occur? What effects do they have? And they might cause what they call copy number variation. And so, a deletion, any chromosomal mutation, is going to depend. What effects it has is going to depend. How big of a region is deleted or duplicated or whatever. In the case of deletions, Well, some of the genes you have are vital. You must have them in order to survive. They encode proteins that are very important metabolically. If those genes are deleted, that's not likely to result in any kind of positive effect. And so the thing is, chromosomes break. Chromosomes can have double stranded breaks. That means both strands in the two strands of DNA are fractured. And usually, if a piece of DNA is not connected to a centromere, it's degraded. And so you just lose some portion of the chromosome. It just depends upon where it has broken. And chromosomes break because, well, accidents happen. Some things, like X rays and gamma rays, for example, have enough energy that if they strike a chromosome, they might break a chunk of it off. Now, if deletions have an effect, they are usually negative. And so there is a human disorder. It's called Friedreich syndrome. And it has been traced down to a deletion. And a little piece of chromosome 5 is missing. And the abnormalities are specific. And they include mental deficiency, a very unique facial structure, and a very unusual, this is how it's got its name. Long time ago, noises that these people made were considered to be cat-like. And that's how that effect, that's how that mutation got its name. And there's, a little piece of chromosome 5 that's just gone. Now, a duplication is going to mean that there is extra genetic material. And it might be caused by unequal crossing over. And so usually crossover events are carefully regulated and they are reciprocal. One chromosome gives up a piece of DNA that's identical to the piece of DNA it gets back. However, there are some places on certain chromosomes where the sequence is very repetitive. And that might cause those two chromosomes to misalign. And that might cause that unequal crossing over, which can usually cause a deletion in one of those chromosomes and a duplication in the other chromosome. Now, duplications are not always bad because sometimes what a duplication will result in is the formation of what they call a gene family. And you might have several genes that encode very similar proteins, but very subtly different proteins. And this can result in things like, as a mammal, as you go through life, you make different types of hemoglobin. And you make fetal hemoglobin, embryonic hemoglobin, not in that order. And you make adult hemoglobin. And the three different types of hemoglobin are caused by you express slightly different combinations of the genes. And that is an evolutionary favorability for mammals that have live young. And kind of like deletions, what effects a duplication has is going to depend upon how big of a piece of that chromosome is duplicated. Now, duplications tend to be less deleterious than a deletion. And that's because you have mechanisms that can adjust the dosage of DNA that you have more than two copies of. And there are some well defined syndromes caused by duplications. And one example is a disease called Charcot-Marie-Tooth disease. It results in, among other things, problems in the nervous system that innervates your extremities. And like I said, duplications might result in the evolutionary formation of gene families. And this allows individual genes in that gene family to accumulate mutations. But since you have a backup copy, these mutations are not always going to knock out that gene. And so, after successive generations, you might result in a gene family where you have multiple genes that have similar but slightly different function. And so I mentioned that you make at least three different types of hemoglobin, and they differ in their ability to bind oxygen. Embryonic hemoglobin has a very strong affinity for oxygen. Fetal hemoglobin is a little bit less. And then adult hemoglobin has the weakest affinity. But this allows the embryo and the fetus to get oxygen from their mother's hemoglobin. Now, specifically, two or more genes derived from a common ancestor are called homologs. If you find these homologs in a single species, they are paralogs, which I always confused, but so be it. An example, like I said, you have the globin gene family. And you have different kinds of hemoglobin. Another member of this family is myoglobin. Hemoglobin carries oxygen around in your blood. Myoglobin stores oxygen in your muscle cells. So when your muscle cells start contracting, when you start exercising, there is a slight store of oxygen within the cell itself. And as I mentioned, you have embryonic, fetal, and adult hemoglobin. There is now, as of within about a year or so, you might have a disease, sickle cell anemia. And if you inherit two copies of a mutant allele that causes changes in the beta globin protein chain of your hemoglobin, you have sickle cell anemia. And you don't really want sickle cell anemia. But there is now an operation that can go in and reactivate. And so instead of making adult hemoglobin, you start to make fetal hemoglobin again. And the thing is, fetal hemoglobin doesn't have that protein chain. And so you no longer have that disease. But there will come a time that if you are a female, at some point in the pregnancy, your red blood cells and the fetus's red blood cells that you are carrying will have the same attraction for oxygen. And I am sure that there is something that can be done to allow the fetus to continue to pull oxygen away from the mother. What that is, I'm not going to make any kind of guesses. But here is some of the globin gene family. You have a bunch of alpha chains, and you have a bunch of beta chains, and different combinations of alpha and beta chains are found in different types of hemoglobin. And then you have one thing over here that's devoted to myoglobin. And then you have copy number variation. And it's just a variation in a piece of DNA that's a pretty fair size. And it has copy number differences between individuals of the same species. Typically, we say that if you are diploid, you have two copies of every gene. However, some individuals might have three or four or however many copies of that gene. Some chromosomes might be missing that gene entirely. Some chromosomes have extra copies of that little piece of DNA. Usually, the copy number variation is small enough that there's generally not going to be any kind of physical consequence. But in some unlucky cases, it causes some sort of genetic disorder. And so generally, there's one copy of an allele per chromosome. However, in sometimes there might be more than one copy of that allele on one of those homologs. And generally, that is fairly minor. Now you have inversions and translocations. And there's two types of inversion, those that span the centromere and those that occur on one side of the centromere or the other. And inversions make some interesting events happen in meiosis. And there's at least two things that can result in translocations. And again, these translocations cause some interesting observations to occur in meiosis. An inversion is just a piece of DNA that is now in the opposite direction. And if I'm face to face, I say basically, this is the order in which the chromosomes should run. And in an inversion, it's broken, but it's been repaired. It's just now in that orientation. A inversion can be referred to as a paracentric inversion, which means the centromere is inside that area that's inverted, or it can be considered a paracentric inversion. The inversion is in one arm or the other. And so this one spans the centromere, and this one does not. And they can have different effects. Usually, in your somatic cells, inversions are kind of harmless. However, if the inversion happens and the breakpoints were right in the middle of a gene, that gene is destroyed. Alternatively, sometimes when you have an inversion and you put the genes in a different order, different geographical regions of a chromosome might have differing levels of gene expression. And it goes on to say a small fraction of the human population carries an inversion that you can see. And mostly they don't know it. But there are a few cases where it will show up. And so usually you can assume that if an inversion occurs on one chromosome, its its homolog is normal. And that results in some weird gametes being made. And that's because crossing over happens in meiosis. It rarely happens in mitosis, but it always happens in meiosis. And that inverted sequence causes some misalignment between the homologous pairs. And so during meiosis one, homologous pairs line up next to each other. And they line up next to each other based upon a sequence similarity between the two chromosomes. But if one of those chromosomes suddenly has a big chunk of DNA backwards, the chromosomes involved in that pairing have to do something to make their sequences align. They have to form what is called an inversion loop. And this might result, if crossover occurs in that loop, it might occur in what they call a dicentric chromosome. Chromosome has two centromeres, and that's not good. Or it might result in an acentric, you can't use the word chromosome, it might result in an acentric fragment, piece of DNA with no centromere. And your cells detect that piece of DNA, and they detect it as DNA damage, and they destroy it. But here's the region of the inversion. And all of a sudden, these things don't align. And so to make these things align, you're going to have one of these chromosomes loop. And now D and E and F and G match up with each other. And if crossover occurs right there, well, you get one basically normal chromosome. You end up with one chromosome that has a deletion and a duplication, so they're abnormal. And then you have that last chromosome again is pretty much normal. And if you have that paracentric inversion, again, it's going to form a loop. Again, things now align. And you'll end up with, again, one chromosome that's essentially normal, another chromosome essentially normal, but then you'll have this fragment with no centromere, and that'll get broken down. And you have this other thing with two centromeres. And what's going to happen is spindle fibers will attach to both centromeres, and that chromosome will be broken when the chromosomes get pulled away from each other. Now, again, we have translocation. Piece of one chromosome becomes attached to something else. Well, sometimes what will happen during this translocation is you'll lose a telomere. And that tends to cause the chromosome that has lost its telomere to be degraded. You might have a reciprocal translocation, which you keep the telomeres. But now you have pieces of DNA that are on different chromosomes than they should be. And again, no DNA is missing. It's just somewhere else. Now, again, reciprocal translocations can result from chromosome breakage and then errors in DNA repair mechanisms. And every now and then, non homologous chromosomes cross over with each other. And so these reciprocal translocations rearrange the structure. They change where the genes are, but they don't cause loss of any genetic material. And so they tend to be referred to as balanced locations. Now, in your somatic cells, these are usually not going to have any major consequence. Every now and then, those translocations can result in a position effect and change the level of gene expression. And that can cause problems. And then you can have what they call an unbalanced translocation. Pieces of the genetic material are duplicated or deleted. These unbalanced translocations cause a change in copy number of the genes. And that can have severe effects, severe disorders, or even death. Now, there is actually a translocation has been given a name, and it's fairly common. 1 in 900 births. And what has happened in this translocation is a bunch of chromosome 21 is attached to chromosome 14. And what is going to happen is that at the extreme end of chromosome 21 and chromosome 14, they're prone to breakage. And sometimes little pieces of DNA with no centromere get lost. Larger fragments might fuse together, and they make a very specific, abnormal looking chromosome. And so you will go from this normal combination to a chromosome containing long arms of chromosome 21 and 14, and one containing the short arms of those two chromosomes. And so there's some DNA that's been misplaced. This one is usually going to be broken down. Again, if you have a translocation, it may or may not have any effect on somatic cells, but it is going to have an effect on the formation of gametes. And in the case of an inversion, For the homologous chromosomes to line up properly during meiosis one, they have to make an inversion loop. In the case of a translocation, what has to happen to make the chromosomes align correctly is you have to form what they call a translocation cross. And that has some troubles that might result when the chromosomes segregate into different cells. And there are three kinds of things: alternate segregation, which is balanced. And then there is adjacent one segregation. And there is adjacent two segregation. And the adjacent patterns lead to chromosomes that are going to have duplications and deletions. Adjacent to segregation is the most rare, but alternate and adjacent one are basically about equally likely to happen. And so here is the cross. The normal chromosome, in this case one, in this translocation. So here's chromosome 2. It's got a big piece of chromosome 1 stuck to it. And so you have to have these crosses form that allow the chromosomes to align. And in alternative segregation, it's basically going to be normal. In adjacent one, you can look at these cells that are made, and well, these down here have more purple than blue. And these up here have more blue than purple. And the same thing is over here. So all of the gametes are unbalanced. And in the other adjacent segregation, all of the gametes are unbalanced. Now, again, I said alternate and adjacent one are the most common, and they're at roughly equal frequencies. Adjacent to segregation is the most rare. And in either kinds of adjacent segregation, the gametes get produced that are abnormal, and it affects the fertility of the individual that has that translocation and in one of those adjacent forms of segregation. Now, there is a definition of euploid, and there is a definition of aneuploid. And then there's a definition of what they call polyploidy and aneuploidy. Euploidy is the normal combination of chromosomes you find in an organism. And so an organism can be diploid, 2 copies of every chromosome. It can be triploid, 3 copies of every chromosome, tetraploid, 4 copies, etc. If you have 3 or more complete sets of chromosomes, the organism is polyploid. And aneuploidy is when you do not have a complete extra set of chromosomes, but at one chromosomal position, you have an abnormal number. 3 copies of the chromosome or 1 copy of the chromosome. Trisomy or monosomy. No, this is a trisome. Yeah, a triploid. This is tetraploid. This has one extra copy of that chromosome, number two. And this one has one too few copies of chromosome number one. Now, in animals, polyploidy is usually detrimental. There are some exceptions of animals that naturally have more than two copies of every chromosome. Plants seem to tolerate polyploidy very well. Aneuploidy is almost universally bad. And in aneuploid, you have variation in the number of a particular chromosome. And it almost always causes phenotypic abnormality. And the easiest way to explain it is that there is a dosage imbalance. And so you overexpress the genes in that chromosome that you have three copies of, or you underexpress the genes in that chromosome you only have one copy of. And so usually these misdosages are lethal, actually. Now, even though aneuploidy is virtually guaranteed to be a lethality, it's not that uncommon. And it is estimated maybe 50% of all human spontaneous abortions are due to aneuploidies. And that's a shockingly huge number. Now, some of these aneuploidies are tolerable. And the one that is most compatible is trisomy of chromosome 21. It causes Down syndrome. Other survivable aneuploidies are trisomy 13 and 18. Much more rare and much less survivable. Aneuploidies at the sex chromosomes, much smaller of a deviation from normal phenotype. Now, you have to have one X chromosome. But if you have more than one X chromosome, your cells are going to deactivate all but one. That deactivation doesn't happen immediately, but it will happen. And so if you are, for example, XXY, you're going to inactivate one of those X chromosomes until you only have one. Now, you're not going to inactivate that chromosome right after fertilization. It'll take a while. And so those genes on that X chromosomes are being expressed for the first couple of 3 months. And so you have too many of them. And so that causes some problems. And, these are some numbers. Trisomy 13 is incredibly rare. Trisomy 18 is very rare. And when it says here, early death, early means before 3. Trisomy 21 is shockingly common. But it does cause characteristics. And I'm sure almost everybody has met somebody with Down syndrome. And I'm sure you're all familiar with the physical characteristics. And then you have the aneuploidies of the sex chromosomes, XXY, XYY, XXX, or X0. These first three, not really that uncommon. And they all have names. And they all have some characteristics. And then you have X0, much more rare. Has a name, Turner syndrome. And a much more abnormal characteristic. And the abnormalities in chromosome number tend to be caused because the gametes themselves don't express their own genes. They are basically nursed by surrounding cells that are almost certainly going to be euploid. And so the gametes that are made can have abnormal chromosome number and contribute abnormal chromosome number to the zygote. Now, some human aneuploidies seem to be influenced primarily by maternal age. And so the likelihood of an aneuploidy occurring begins to increase fairly rapidly after about the age of 40. It is also affected by the age of the father, but nowhere near as significant. Now, in Down syndrome, which is the most common, it is caused by the failure of chromosome 21, most likely to not segregate properly. And in the case of at least a primate, the oocytes, those cells that are going to produce the eggs, are made before birth. And then they go basically into storage. And at puberty, that phase, that meiosis restarts. And it seems that the longer that those oocytes are stuck in prophase 1, the more error prone they are when they resume meiosis. Now, there are examples of animals that have variation in euploidy. And there's also what they call endoploidy. And there's also this really interesting thing called a polytene chromosome. And then there's polyploidy in plants. Euploidy is variation in the number of complete sets of chromosomes. Most animals are diploid. They have two. In most animals, deviation from diploidy is not a good thing. And polyploidy in the mammalian family is generally lethal, although there are exceptions. And in some organisms, for example, bees might experience haplodiploidy. Female bees are diploid. They result from a fertilized egg. Male bees are monoploid. They result from an unfertilized egg. And there are some examples, fairly well studied, where polyploid animals have been found. And this is a type of frog. And I believe that this one is a diploid animal. And this one is a tetraploid animal. By definition, these two animals are reproductively isolated from each other. Therefore, they're of different species. And then you have endopolyploidy. Certain cells in certain types of tissues might be polyploid. For example, it is not unusual for cells in your liver to be tetraploid, triploid, or tetraploid, or even more. And then you have these things called polytene chromosomes. You usually find them in insects. And in a polytene chromosome, you have a chromosome that has certain regions on it where at certain points of development, you need lots and lots and lots of copies of the gene products in that area of the chromosome. And so you can overcopy that little region of the chromosome and make it huge. And it's pretty easy to see microscopically. And so basically, little areas of the chromosome undergo repeated rounds of chromosome division, but then the cells don't divide. And in fruit flies, chromosomes might double 9 times. And those doublings produce a big bundle of chromosomes that you can see. And that big bundle is called polytene chromosome. And that vaguely central point, where all of that stuff comes together, is called the chromocenter. And there's the action of just copying over and over and over and over again. And it's not followed by cell division. So they all stay stuck together. Now, polyploidy seems to be fairly common in plants. And they say about a third of flowering plants and ferns might be polyploid. And we humans, have done this during crop domestication because polyploid plants tend to have bigger fruits and bigger seeds. And that is often the plant material we eat is fruits and seeds. And so these polyploid plants tend to be bigger and have a bigger seed set and just healthier. But polyploidy in animals, not a good thing. In plants, seems to be okay. Now, if you are a polyploid organism and you have an odd number, that means you're a triploid or pentaploid or whatever. Usually, those organisms that are triploid or pentaploid or heptaploid, I believe is the prefix for 7, are sterile. And what happens is during gamete formation, there's all of this unequal separation of chromosomes. And that tends to make inviable gametes. Now, sometimes that sterility is desirable. We have made seedless fruit. And for example, watermelons and bananas and grapes are triploid or something. They cannot reproduce on their own. And so humans have to make those things reproduce. Some flowers are seedless, and we view that as a good thing. And let's see. I am not going to finish this today. I thought I could. It's been an hour. I don't have long to go. But there are various mechanisms that can produce variation in chromosomes. And there can be all kinds of different non-disjunction errors that might happen after certain events and might change the phenotype in specific ways. And there's something called autopolyploidy, alloploidy, and allopolyploidy, which we explain in a little bit. And you can introduce a drug to a lot of plants that causes them to be polyploid. And there's a drug that is in common use. It's called colchicine. And it causes a complete non-disjunction error. So the gametes that are made are, for example, diploid. And so when two diploid gametes fuse, you make a tetraploid organism. Now, that is where I'm going to stop today. When about the question at noon, when I post the exam, I have to put in an available from date and a due date and an available until date. And so when I get all that set, I will make an announcement. Some people, some students, for whatever reason, did not take exam one. And I have it set up in this class that you don't get to drop an exam. And so I have to do something for anyone that didn't take exam one. I have to somehow allow them to take exam one. Now, anyone that falls into that category at midterm, you will have a poor grade, very likely an F. But midterm grades don't really mean anything. So you should know that there are lots of points still out there. And I am going to have to give you a second crack at exam one. And so your grade from point of midterms, which are due, I have it written down, it looks like March 17th. Between March 17th and when final grades are due, which is May 12th. And I just looked, and I have a doctor's appointment on May 12th, which means I have to put your grades in no later than May 11th. Between middle of March and beginning of May, there is going to be a vast potential for your grade to change. And so just keep that in mind.